Hello guys, welcome to Intellect Medical ZM. So as promised, today we begin our discussion on HIV and we'll be looking at part 1, which is definition of terms. These are basics, but remember basics are important in understanding topics because in certain topics there are certain terminologies that are used and they are quite unique to the topic, and for you to really understand what is being talked about, you first have to understand what some of those terminologies mean. And not only that, but in certain conditions such as HIV, certain terminologies will help you to understand why certain things are done in a certain way and why certain things are important or why certain things need to be monitored, stuff like that. Yeah. Okay. So since this discussion is about HIV, so first things first, let's briefly talk about what HIV is. So HIV simply stands for human immunodeficiency virus. So basically, what the HIV virus does is in what these same letters stand for, it causes some sort of immunological deficiency. So, this immunological deficiency comes about in the sense that the HIV virus, destroys a certain type of immune cells that helps in mounting an immune system. An immune defensive response towards a pathogen. Yeah, and by doing so, it makes now the body vulnerable to certain infections. Yeah, okay, and it is important to understand that HIV may progress to a life-threatening condition which is known as AIDS. Yeah. So AIDS stands for acquired immune deficiency syndrome, and this syndrome comes about when an HIV infected individual begins to present with opportunistic infections and some oncogenic viral associated tumors. Yeah. So these infections are occurring because the immune system has been damaged, or rather, uh, certain cells of the immune system that are important in fighting infections have been destroyed. Hence, the body is now vulnerable to these infections. Yeah, and that's what AIDS is all about, where someone begins to present with different kinds of conditions as a result of the impaired immune function. Yeah. Okay, so now we talk about viral load. So viral load simply refers to the number of viral copies that are present in blood. Yeah, so remember that when the virus enters the body, it begins to replicate, making copies of itself. Yeah, so I've already even explained what viral replication is all about. It's just making copies of itself. So as it begins to make copies of itself, it begins to increase in numbers. Yeah, so such can be measured using blood. Yeah, and that's what viral load refers to. Yeah. Okay, so CD4 count. Before I talk about what a CD4 count is, which is of course a measure, I want you to understand something. A CD4, which simply means cluster of differentiation, it is a glycoprotein or a receptor that is present on certain types of immune cells. Yeah, and these are the same immune cells that are targeted by the HIV virus, and it includes cells such as the monocytes, macrophages, the dendritic cells, yeah, and the T helper cells, yeah, which are a special kind of T lymphocytes. Yeah. So CD4 count is now a measure of these same cells of the immune system that are targeted by HIV in blood. And according to the World Health Organization, it is said that in an adolescent and adult, the normal range should ranges from about 500 to 1,500 cells per milliliter. Yeah. So that's it about CD4 count. The other thing that I want you to understand about these two parameters, viral load and CD4 count, is that they are used to monitor disease progression. Yep, because viral load gives you a picture on how much damage the virus has caused to the immune system. And the CD4 count actually just gives you a picture on uh, the number of CD4 count that are remaining now within the blood. Yeah. So the lower the number, the higher the risk of developing opportunistic infection. So these are actually important parameters that needs to be monitored from time to time to check how one is, perhaps responding to treatment. Yeah, stuff like that. Okay, so we proceed to window period. So window period, this one simply refers to a time period between exposure to the infection and the time when the body develops antibodies against the HIV, or the the time period between exposure to the infection to the time when an individual tests uh, positive. Yeah. So now, the other term that we'll look at is seroconversion. And the reason why I've placed them like this, is because oftentimes people use them interchangeably. Yeah. Sometimes someone would use seroconversion to mean window period, but here's something that I want you to get that helps you differentiate the two. Window period is the time when you've been exposed to the infection, but you test negative because your body has not yet produced antibodies against the HIV virus. Yeah. Whereas seroconversion mainly focuses on the appearance of anti-HIV antibodies in your blood. Yeah. That time period when now you get an accurate positive result. That is what seroconversion is. And if you were to describe window period in other terms, you would say it's a time period between exposure to the infection and before seroconversion, because that time when you've been exposed and you test negative is what window period is all about. But the moment you test positive, that's seroconversion, because now your body has produced enough antibodies towards the HIV virus, and now they are detected accurately with a positive result. Okay, the other thing that you need to be aware of is that there's another the terminology that's used, which is incubation period. So don't confuse the two, because incubation period mainly focuses on from what time, or to what point did this individual show signs and symptoms. So yes, you've been exposed. Let's say yes, you've been exposed to the coronavirus. From the time you were exposed, to what point did you begin to show signs and symptoms? That's what incubation period mainly focuses on. Meaning, during that incubation period, you may actually even show positive, but you may not have the signs and symptoms. So something like that. That's a picture you should just have to maybe try and differentiate the two. Yeah. So viral replication, I've already said what it's all about. It's all about the virus making copies of itself, yeah, using the host cell genome. Yeah. Okay. So proviral DNA. Okay. So this proviral DNA is actually when now the RNA, the viral RNA, has been transcribed into the viral DNA, and it has been integrated into the host cell genome, or into the host cell genetic material. That is now what's referred to as the proviral DNA. When the viral DNA is integrated or inserted into the host cell's DNA. Yeah. Okay, so genome, I've already used it in some other descriptions up there. So it simply refers to the genetic material. So whenever you hear genome, just think of things like DNA, genome, DNA, genetic material. That's it. And host cells, basically it just refers to self cells or body cells. Yeah. So when you hear something like the host cell genome, it's about the body cells genetic material, something like that. So those are things that you just need to understand in order to avoid being confused on what's really being talked about. Yeah. Okay. And the other terminologies that we'll look at is transcription and translation. So in simple terms, transcription simply refers to copying DNA into RNA. That's what transcription is. And translation, in simple terms, is just the copying of the RNA into proteins. That's translation. Yeah. But basically, because in the next presentation that we'll do about the pathophysiology of HIV, we'll talk about some of these things, and that's the reason why I thought it's important to mention really what these terms mean, so that in case we don't say much about them in the next tutorial, at least you understand what we mean by transcription and translation. So, in other words, if we're to become a little bit scientific, is that transcription, this is where now when the body copies the information on from the DNA. Yeah. So it copies the information on one strand of DNA. Yeah. And forms an RNA. Yeah. An RNA base, yeah, or component. And this RNA component is actually just containing information on some proteins, or rather polypeptides, that are to be formed. Yeah. Which is simply referred to as gene expression. So when when the when the enzyme RNA polymerase begins to copy the information from the DNA and forms this RNA, it will just contain information on which proteins to be formed. And later on, this same RNA will be translated, meaning now that information that is within the RNA will be interpreted to now produce the exact protein that is needed. Yeah. And you understand more about this when we get to talk about the pathophysiology. Yeah. And basically, remember, genetic expression, or rather when we talk of copying of information, interpreting of information, this information in the DNA as well as the RNA is just stored in the form of bases. You remember those things, adenine, guanosine, yeah, cytosine, things like that, thiamine, yeah, uracil. So those are like bases, yeah, that are put together to form like nucleosides, some stuffs like that. So the same bases, yeah, will be uh, copped. They'll be interpreted and they'll be copped to form this RNA. And this RNA, when forming proteins now, is that the bases are grouped into three pairs, and this is what is referred to as a codon. So these three pairs, each three pair uh, contains information on a certain type of amino acid to produce, and these amino acids, when put together, they will build now this polypeptide, which is a protein molecule. Yeah. So I don't want to get into details, but I just thought that it would be better to just tickle your memory a little bit, so that you can begin to they'll think deeper as you think of these terminologies, transcription and translation. Well, I think that's it about the presentation on terminologies. I hope this helps, and looking forward to working on another presentation on the pathophysiology of HIV. Yeah, thanks for your time.